Now, some of you, oh, recording in progress, we're good. Now, some of you might not know a lot about Mount Allison, so I thought I'd take a second to tell you about it. Mount A is a primarily undergraduate institution located right across the Nova Scotia border in Sackville, New Brunswick. This image that you're seeing of Mount A here is the picture that the administration loves to include on their promotional materials for Mount A, probably because it's the view of campus from the administrative buildings. But I like this picture a lot because in it, you can see my lab. Let's see if I can make this work. Yeah, my lab is located right behind the two windows here in Flemington building, and you can see the greenhouse. So those are the locations on campus where I do my research. And of course, I have field sites located around New Brunswick too. Both my research on campus and my research at field sites in New Brunswick takes place on the unceded territory of the Wolastoqiyik and the Mi'kmaq peoples. As a plant evolutionary ecologist, I'm interested in the interactions between plants and their environment and with other organisms. But my Western science way of understanding these topics is just one way of understanding the world. And of course, the first peoples of this land hold a deep knowledge of plants and their relations to other beings. As a plant evolutionary ecologist, I do what I do because I think plants are fascinating. And plants interest me for a number of reasons, but the one I want to focus on today is the way that plants grow. Plants grow by adding new parts to their body. And they can do this because at every shoot tip, every root tip, and every leaf axil, a plant's got a cluster of undifferentiated or largely undifferentiated pluripotent cells called the meristem tissue. Cells in the meristem, oh, I hope you can see my cursor here. Cells in the meristem can divide and produce new organs. So through meristematic divisions, a plant produces new roots, new shoots, new leaves, new flowers. And because plants grow through this continual addition of new parts to their body, it's for that reason that when we look at a young plant, like the oak seedling over here, we can't fully predict what the individual is going to look like when it's a mature individual, like the mature oak tree shown here on the right. Plants are continually changing the structure and composition of their body. Of course, there is some growth through the expansion of existing parts. In plants, you get growth in the internodes between leaves, and you get expansion of the girth through the vascular cambium. But the majority of plant growth takes place with the addition of new body parts. And that's quite different than the way that animals grow. In animals, the body plan is laid out early in development, and the individual grows by expanding the size of those existing body parts rather than adding new body parts to themselves. That difference can be summarized in a more formal way by saying that plants are modular organisms and animals are unitary organisms. I put the most in parentheses here because, of course, like everything in biology, there are some interesting exceptions to these rules on both sides. But we're going to focus on the general case that most plants are modular, most animals are unitary. And it's the difference between growing by adding Legos to your Lego tower versus growing by inflating the balloon. And that plant mode of growth by adding pieces to the tower has some interesting consequences for the way that plants interact with their environment, right? As a plant adds new pieces to its body, so new bricks, or in the example here, new leaves, when one leaf is added, it's not going to be a perfect copy of the leaf that came before or a perfect copy of the leaf that will follow next. In this illustration here, the leaves on this pothos vine might differ in their exact shape, in their size, in their thickness, in the amount or distribution of the yellow splotching on the surface. And so we see that there's variation from organ to organ within the plant. This is called the within plant variation, right? It's variation among the repeated organs within a plant. And within plant variation can be surprisingly large. To help illustrate what I mean by this point, I've got a little cartoon here. So we're imagining a population of 4 plants, and our plants are producing flowers that are represented by the purple circles here. What I hope you notice is that there's some variation in flower size within this population. So I could break out some sort of a ruler and measure flower size. We'll go ahead and do that now. And then on whatever my arbitrary scale of measurement is in this example, we see that some flowers are of size 3, some are of size 2, and other flowers are of size 1. Now that we've measured all of the flowers in the population, I can go on to look at the flowers presented by each individual plant and calculate the mean and the variance of flower size within each plant. The way that I've created this cartoon here, the plants all have a mean flower size of around 2, and the variance in flower size differs a little bit from individual to individual. With our within plant mean data, we now go on to calculate the variance in average flower size across individuals. So I take my means and I calculate the variance of those means. Again, that's the variance in average size across individuals. And we see that we're getting a very low value. And that value of the variance in mean flower size across individuals is very small compared to the variance in flower size that's occurring within individuals, right? So in this cartoon, within plant variation is very large relative to the variation in organ level traits that's occurring across plants in this population. So it's easy to make variance partition the way I want it to in a little cartoon like this, but how prevalent is this phenomenon in the real world, right? How often does variation in organ level traits within plants overwhelm variation among plants in those same traits? Well, in a 2009 book, Carlos Herrera asked exactly that question, and he assembled as much data as he could on measurements of organ level traits from the literature, and wherever that data was collected in such a way that he would be able to partition the variance to variance occurring within plants versus variance among plants. We can see the result of his large assembly of data in the slide here. And what we're seeing on the y-axis is for any given trait, what proportion of the variation in that organ level trait was occurring within plants. The proportion within plants is equal to, or the proportion among plants is going to be equal to 1 minus the variation that's occurring within plants. For traits that were associated with leaves, so things like leaf size, leaf shape, the variance occurring within plants was extremely high, right? The median value here across 23 traits was about 75% of the variation was occurring within plants. The variance within plants was also high for traits related to seeds, traits related to fruits, and even for traits related to flowers. And flowers are, structures that we expect to be pretty conservative in their construction because they're so important for plant mating. But even for flowers, the median value was about 40% of the variance in a floral trait was occurring within individuals rather than among individuals within a population. So variance within individuals is large, and it's surprising to me then that it receives maybe less attention relative to its magnitude in the study of plant ecology than you might expect. You might say that plant ecology often neglects the potential importance of within plant variation. Where within plant variation is considered in plant ecology, it's often from one of two perspectives. One dominant view on within plant variation in the plant ecology literature is that within plant variation is an impediment to selection on organ level traits. That view is true, right? There's absolutely merit to this argument, and I'll elaborate on that point later on in the presentation. A second dominant perspective on within plant variation in the plant ecology literature is that within plant variation is a nuisance. And that's also true because it's one more thing you have to account for, one more thing you have to take into consideration in your experimental design when you're working with plants. So both of these perspectives are right, I don't disagree with either of them. But I do wonder if maybe it's not the whole story on within plant variance. If within plant variation is where most of the variation in organ level traits is lying, maybe there's something more to it. Maybe the ecological and evolutionary significance of that variation deserves a closer look. And so the potential ecological and evolutionary significance of within plant variation is an idea that just seems to keep cropping up in my work for a while now. And so I decided to make that the theme of my talk for you today. And for the remainder of this talk, I'm going to walk you through 3 possible reasons why I think within plant variation might matter. These are ideas that I have worked on, the first one here, ideas that I'm currently in the midst of working on, and ideas that I've only just started working on at the end. So you'll see as we go through the talk, the amount of information I have to provide you in terms of data on these 3 topics decreases over time. But yeah, the first idea is that maybe within plant variation is an agent of selection. Second, maybe it's a target of selection. And third, maybe it could be a blueprint for diversification. Right. And so I think we'll go ahead and start talking about these 3 topics one at a time. So the first thing I'd like you to think about is the possibility that maybe within plant variation could be an agent of selection. Now remember, selection is simply an association between a trait and fitness. And an agent of selection is some component of the environment that causes the association between trait and fitness. So what this statement here is saying is that maybe within plant variation expressed by one individual is shaping the environment and the fitness opportunities experienced by other individuals in the same population. The particular example with which I've been developing this idea has to do with within plant variation in fruit set probability and how changes in fruit set probability within one plant shape the mating opportunities and the male fitness prospects for other plants in the population. So what do we mean by within plant variation in fruit set probability? Well, as you're probably aware, many plant species produce multiple flowers over the course of a growing season, but those flowers aren't all deployed on the same day. So in the lupins shown here, for example, the flowers at the bottom are wilted. They're old flowers that were open and active before this photograph was taken. And up at the top, we've got the flowers that are still in bud and are yet to open, right? They're going to open it some number of days after this photograph. Now, any flower on this plant in the lupin could, in theory, receive pollen grains from some other plant in the population. And then if its ovules are fertilized, those ovules can mature into seeds, the flower matures into a fruit, right? So any fertilized flower can, in theory, mature into a fruit. But the reality is that even if a flower is fertilized, it's not necessarily true that it will mature into a fruit. The maternal plant has some control over which flowers do and do not mature into fruit. And it turns out that frequently it's the flowers that open first on a plant. So in the case of the lupin here, the flowers on the bottom that are more likely to mature into fruit than are the flowers that open later on the same individual plant. So in this fruiting lupin over here on the left, we see at the bottom of the inflorescence, the flowers have matured into fruit. At the top of the inflorescence, they have not. So the probability of fruit set decline has decreased from the first to last flowers within the individual. That pattern is occurring in lupins, at least in these two photographs, but it's a widespread pattern across flowering plants. This little histogram here is just some data I assembled from the literature during my PhD to try and get a sense of how common it is for fruit set to decline from first to last flowers within plant and how strong that decline generally is. The green line in this histogram is the 0 line, where there would be no change in fruit set probability from first to last flowers. And as you would expect, most of the data sets that I assembled in this non-exhaustive review were showing a negative change in fruit set probability, meaning that the first flowers were more likely to make fruit than the last flowers. And in some instances, in many instances, that change in fruit set probability was quite large, right? 0.75 or greater. So within plant decline in fruit set probability is a widespread phenomenon. My co-authors and I on this paper, so myself, colleague Jessica Forrest, and my PhD advisor, Art Weiss, we're thinking about this within plant decline in fruit set probability and how it might affect the mating opportunities of other plants in the population. And the idea that we came to is that when fruit set declines from first to last flowers within a plant, it might lead to selection on flowering time to the male component of fitness for other individuals in the population. To explain this idea, I need to first make sure that we're all on the same page with what I mean by male fitness in a flowering plant. Probably when you think about fitness in a plant, you think about measuring fitness as the number of seeds produced by an individual, right? Any flowering plant can mature its ovary into a fruit. The fruit contains seeds. Every seed produced by an individual contributes to that individual's female fitness, right? Those are the seeds that an individual produces itself. But the vast majority of flowering plants are hermaphroditic. And so they're achieving fitness not only by producing their own seeds, but also by exporting their pollen to other plants in the population, where those pollen grains can go on to fertilize the ovules of the neighboring plants. And any ovule matured into a seed by a neighboring plant that was fertilized by a pollen grain from our focal plant will constitute that focal plant's male fitness. And whether a flowering plant has produced its own seed or has passed sired a seed on another plant in a population, it's passing on precisely one gene copy to the next generation. And so male and female fitness are equally important. We tend to focus on female fitness because it's easier to measure. But the idea that I was exploring in this part of my PhD was that maybe the within plant pattern of declining fruit set from first to last flowers is going to lead to selection for early flowering through that male component of fitness, right? The success at siring seeds on other plants in a population. Got a lot of cartoons in this part of the talk, but I'm going to give you another cartoon now to help elaborate on this idea, what exactly we think is going on. So this what's the word I'm looking for? This display starts with an axis down at the bottom that represents time in a flowering season, from the first day that the first flower opens on the first individual to start flowering in the population to the last day that there's an open flower in this population. And I'm going to populate the population using bars to represent individual plants. So this bar represents the first plant to start flowering in the population, and it continues flowering until this point in the season. A few days later, a few more individuals start to flower, and then they stop flowering later on. More individuals a few days later, and so on and so forth, until the last individual opens its first flower, produces all of its flowers, opens its last flower on its body, and then that's the end of the flowering season in this plant population. The next thing I want to do is shade in these bars to represent the probability of fruit set across each individual's duration of flower production. So I've gone ahead and done that now. So now the bars are shaded so that the dark part of the bar is when the individual is producing flowers that are likely to mature into fruit. And the open part is when it's producing flowers that probably won't become fruit, right? So that's that within plant variation and fruit set probability. With this shading in place, we can now look at the male mating opportunities of different plants in the population. Starting with the first individual to come into flower, we see that its flowers overlap in time with a number of the other flowers in the population that are likely to mature into fruit. And so from first principles, it should have a high probability of having its pollen grains successfully fertilize ovules that are going to mature into seeds. We expect this individual to have a high male fitness. And I bet you can see where this is going, but if we now look at the mating opportunities of the last individual to come into flower, we see that its flowers are overlapping in time with other flowers that are probably not going to mature into fruit. So it may be successful in sending its pollen grains to those other flowers, but it's kind of a dead end, right? The pollen grains might fertilize ovules, but the plant might not invest in actually maturing those fertilized ovules into seeds. So we expect that last individual in the population to have a low male fitness. The idea works in this little cartoon. In a small paper, my co-authors and I formalized it into a small numerical model that was parameterized with fruit set decline values from that histogram I showed you earlier. And we found that when there's a moderate or strong decline in fruit set probability, we could generate appreciable selection for early flowering through male fitness. All right, so the idea worked on paper, and it led us to create this prediction that because fruit set probability declines within plants, selection through male fitness ought to favor early flowering onset. Importantly, this prediction is saying that selection favors early flowering onset through male fitness simply because the first plant to flower is early, right? It's just the fact of being early that makes it have high male fitness. It's not that there's something intrinsically better about that first plant that makes its pollen higher quality or anything like that. It's just by the fact of being early, it should have higher male fitness. We tested this prediction in some experimental gardens of the model plant Brassica rapa. Brassica rapa was a good species to work with for a number of reasons, but the one I'll just point out to you here is that this species was previously reported to exhibit the required decline in fruit set probability from first to last flowers. So here on the x-axis is the age of a plant at the time that a flower is deployed. And on the y-axis is the probability of that flower maturing into a fruit. And what you'll hopefully see is that the first flowers produced by the plant when it had only started flowering had a high probability of maturing into a fruit, whereas the last flowers were unlikely to mature into fruit. All right, so Brassica rapa displays the required decline in fruit set probability. And so with Brassica rapa, I constructed experimental populations of B rapa, Brassica rapa. And then in those populations, I measured each individual's day of flowering onset, the number of flowers each plant deployed within some defined time interval. You can think of those intervals as how many flowers were produced each day, but in reality, I was measuring time intervals of 3 days or 4 days. Within each of those intervals, what proportion of the deployed flowers actually went on to mature into fruit, right? So here is where I was establishing that pattern of fruit set decline within individuals. I also measured the total mass of seed produced by each individual. That's its female fitness. And then finally, through genetic paternity analysis, I estimated the total seeds sired by each individual plant, which was that estimate of male fitness that I needed to have to test our prediction. It's kind of funny for me to see all of this on one slide because it was a monstrous amount of work. But as you get further in time away from your PhD, I think it becomes easier to just kind of put it in a nutshell. And so that's what I did for the major experiment in my PhD. All right, now remember the idea I wanted to test was that the first plants to flower in a population should have higher male fitness simply because they're first. If I had just tossed seeds into a garden and let them germinate and flower when they wanted to, I would have had a hard time testing this prediction. And that's because there's an inherent correlation between the timing at which an individual starts flowering, so whether it's the first plant to flower in a population or the last plant to start flowering in a population, and the genetic content that individual has that controls the age at which it starts flowering, measured as the number of days from germination to the first flower production. Assuming that there's relatively low variance in germination time, and in all my work in Brassica rapa, there always has been, assuming low variance in germination time, the first plant to start flowering in a population should be one that's carrying genes for flowering at a young age, right? One that doesn't take very long to go from germination to first flower production. And the last plant to flower in a population is one that's carrying genes for flowering at an older age, one that takes more days to go from germination to first flower production. So the problem here is that if I want to test the idea that plants that flower on an early date have high fitness simply because they're on an early date, and I did go on and detect that, in fact, there was selection for early flowering, I wouldn't know if the plants that were flowering early had the highest male fitness because they were early, or because there was some other advantage to being young that I hadn't controlled for. So in order to really isolate the effect of being early from the correlated effect of being young at flowering onset, I had to manipulate that correlation between age at flowering onset and timing of flowering onset. And so I created 3 types of gardens in this experiment. And as I show you the way that these gardens are constructed, I'm going to color code individuals by their genetic character for age at flowering onset. So the points that are purple are the plants that are carrying genes for flowering at a young age. The points that are pink are the ones that are carrying genes for flowering at an old age. And then you can see other ages of flowering in the other colors here. So the first type of experimental garden I created maintained that intrinsic natural correlation between timing of flowering onset and age at flowering onset. So the first plants to flower flowered young, last plants to flower flowered old. In another treatment, I reversed the correlation so that the first plants to flower in the season flowered at an old age, and the plants that flowered last in the season were ones that flowered at a young age. So they took the least time to go from germination to first flower production. And in the third treatment, I disrupted the correlation so that there was no association whatsoever between the timing at which an individual started flowering, was it early or late relative to other plants in the population, versus its age at flowering onset, the number of days it took to go from germination to first flower production. I achieved this manipulation by rearing Brassica rapa in the greenhouse for 2 generations prior to the onset of the experiment, and keeping track of each family's age at flowering onset, so that after those 2 generations, I could take a seed from a known family and predict with reasonable accuracy what the age at flowering onset of that seed would be. And then with that knowledge, I could stagger the planting dates of seeds from different maternal families to create the correlations that I was after, right? Once I had these gardens established, I could make some predictions about what I would expect selection on flowering onset to look like if the prediction that declining fruit set is shaping male mating opportunities held true. So in the treatment over here, if there was some advantage to flowering early in a population, I would expect that selection favors flowering young because the plants that flower earliest are the plants that flower youngest. In the treatment on the other side, I would expect selection to favor flowering old because the plants that flower earliest are the plants that flower at the oldest age. And in the middle, I would expect to find no selection on age at flowering onset because there's no association between a plant's age at flowering onset and whether it was early or late in the season. So I'm going to show you the results of this experiment on the next few slides, but I want to first introduce the symbols that I'm going to use to represent these 3 treatments. The upwards pointing one is showing us the positive upwards association between age at flowering onset and timing of flowering onset. The downwards triangle is showing us the negative association treatment. And the circle is showing us the no correlation treatment. Right, the first thing I want to show you is just a proof of concept to show you that that age at flowering and mating opportunity were related in the way that I expect it to be, given that fruit set is declining within plants. So all I'm showing you here is that in the positive correlation treatment, represented by the upwards triangle, there was a negative association between an individual's age at flowering onset and its mating opportunity. What that means is that plants with the smallest age at flowering onset, so the youngest age at flowering onset, had the most mating opportunity in this treatment, which is exactly what I expected to be true. In the negative correlation treatment, we see a positive association between age at flowering and mating opportunity. And again, that's saying that plants with the largest age at flowering, i.e. the ones that flower old, have the most mating opportunity. And in the uncorrelated treatment, that association was not significantly different from zero. So these results here are simply showing that the concept made sense, right? In terms of which flowers which plants interacted with the most flowers that actually matured into fruit. Mating opportunity was structured in the way that I expect it to be based on the numerical modeling, et cetera, that I had done with my colleagues previously. So if that distribution of male mating opportunity was the thing that determined male fitness for plants in this experiment, when I went on to look at the association between age at flowering and male fitness, i.e. when I went to look on at selection through male fitness on flowering time, I would expect these points to be distributed in the same way. You can probably already tell from my tone of voice what actually happened. The prediction didn't hold out. Instead, what I found was that in 2 of the 3 populations, there was strong selection for a young age at flowering onset. And that selection was particularly strong in the uncorrelated treatment, where age at flowering had no association with whether a plant was early or late. So what that was telling me was that there was something else about being a young age at flowering and an old age at flowering that was affecting an individual's male fitness. Plants that flowered at a young age were just somehow inherently better at fertilizing ovules than were plants that flowered old, even when the opportunity for fertilizing ovules was removed, right? So in other words, this experiment detected a strong effect of age at flowering on male fitness that overwhelmed any effect of access to mating opportunity that I was trying to test. Okay, so the question I was asking is within plant variation an agent of selection on flowering time? The numerical modeling and simple diagrammatic cartoons showed that in theory, yes, it could be. Within plant variation and fruit set probability could drive selection for early flowering through male fitness. But in my field experiment with Brassica rapa, it was not, right? There was something else about being a plant that flowers at a young age that gave a male fitness advantage that was independent of any association between age and timing of flowering onset. Don't despair for me. That experiment was a ton of work. This was only one prediction that I was trying to test in the experiment. Most of my predictions ended up being wrong for this experiment. But I did learn a lot about the nature of selection on flowering time through this work. And if you're interested in that topic more broadly, these are some of the papers that came out of that project. The other reason you shouldn't despair for me is that just because the prediction didn't hold out in these highly manipulated gardens with Brassica rapa doesn't mean that it has no value whatsoever, right? We know very little about the nature of selection time on flowering time through male fitness. And it could be that as we start to estimate selection on flowering time through male fitness in other plant species and other environmental contexts, maybe this idea that within plant variation is driving selection could hold out, right? So I would say that the idea has merit in theory, and maybe we need more empirical data to figure out whether or not it plays out in the real world. All right, so we're going to move from thinking about whether within plant variation could be an agent of selection to thinking about whether it could be a target of selection. So rather than being the thing that changes the environment to drive selection on other traits, is within plant variation itself affecting the fitness of an individual? Now, I said earlier in my talk that within plant variation is often viewed as an impediment to selection on organ level traits. And the reason why that's true is pretty clear. You can take a look again at this cartoon that we had earlier, but we've got a lot of variation in an organ level trait, i.e. a lot of variation in flower size, but there's not a lot of differences among individuals in their average flower size. So if selection were to act to favor plants with a larger flower size versus plants with a smaller flower size, that selection wouldn't be very effective in this population because there's simply not a lot of variation in that trait. That point is true, right? Having a lot of within plant variation in a trait is going to reduce the efficacy of selection on the mean trait value. But what isn't elaborated on in that point is that the plant mean for an organ value trait is not the only way to characterize that plant. Plants are also characterized by their variance in organ level traits. So in this population, we're seeing that there's actually a lot of differences in the variance and within plant variation and flower size. So the variance in the way that I've drawn the cartoon here ranges from 0 for this plant who produces flowers of size 2 only, up to a high of 1.33. So it could be that maybe it's not the average flower size that an individual produces that affects its fitness, but maybe it's how variable the individual's flower size is. Maybe there's some advantage to having a lot of variation in flower size within the individual, or a disadvantage. Another possibility is that maybe it's not just the overall variation that is affecting fitness, but the pattern in which that variation occurs. So some of my plants here have a decreasing trend in flower size, others have an increasing trend, and others have no discernible linear trend in flower size. And maybe selection could act on those sorts of differences in the way that within plant variation is expressed. And so that's the idea here. Could within plant variation itself be a target of selection? Could it be a trait that affects a plant's fitness? There are a handful of studies where investigators have detected selection on within plant variation, even in the absence of selection on the plant mean value. My favorite of those is work by Mason Kolbaba and colleagues who worked with Delphinium glaucum, the plant shown here. And in a careful study with this system, Kolbaba and colleagues showed that for the first flowers produced on an individual plant, selection tended to favor a larger size of sepals. The sepals form the landing pad on these Delphinium flowers for pollinators to land on. So on the first flowers produced, there was some advantage to having a large sepal size. The same was true for the last flowers produced on an individual plant. Selection favored having a large flower landing pad size. But in the middle of the plant, there was some selective advantage to having a smaller landing pad size. And so this study was suggesting that selection might favor a particular pattern within plant variation. It might be beneficial to have a big landing pad at the bottom and the top, but a smaller sepal size in the middle. So that's a nice example of how selection could potentially act on the pattern within plant variation. But for one, those examples are few and far between. And two, I've yet to see a strong evidence that if selection is acting on those traits, those within plant variation traits, that a population can respond to that selection. And so those are ideas that I'm trying to tackle with my students as we develop a new system for studying within plant variation, Halenia deflexa, which is a member of the gentian family. The reason I've chosen to focus my work on this topic on Halenia deflexa is one, because it's a native annual plant. An annual plant offers a lot of advantages when you're trying to study processes over generations. And Halenia deflexa is kind of a rare bird in our part of the world because it's not often that you find a native annual plant where we live. The other reason I chose Halenia is because its flowers are so darn interesting and there's very little written about the species in the Western scientific literature. One of the things that makes its flowers so cool to look at is these long nectar spurs. Somehow that ended up misplaced. That should be underneath the spur, which is the part that I'm highlighting right here. The nectar spur is a projection off the petal of the flower. It's a tube, and at the base of that tube is where there's nectar located. So the spur affects the mechanical fit between a plant and a pollinator visiting that plant. And so this is a trait that's intimately tied to a flower's reproductive success. What's interesting about Halenia deflexa is that the gray literature about the species, so things like non-peer-reviewed species descriptions, talk about the spur size of the flowers varying a great deal from flower to flower within the plant. So there was this hint that maybe there's some important within plant variation going on in the species about which very little is known, right? So I've had several honor students now helping me to start up this plant species as a possible system for studying the evolution of within plant variation. Right. The first thing my students and I wanted to do was to arrive at a more formal quantification of how much within plant variation was happening in nectar spurs. An honor student from 2019, Roisin Kiersted, who's pictured here, helped me tackle this question. My field crew that summer, including Roisin and myself, traveled regularly to a wild population of Halenia deflexa that occurs along the Nipisiguit River here in New Brunswick. And on each site visit, we returned to the same plants and collected some currently open flowers from that plant to preserve them in ethanol. We then, later in the year, took out those preserved flowers, and Rasheen dissected them, imaged them, and took measurements of organ level traits in the flowers. So the first thing we wanted to do was ask, how much is spur size changing from flower to flower within the plant relative to the day of flower collection? So 0 is the first day that a flower was collected from that plant, up to X number of days later, so 30 days later. She performed this analysis using 86 flowers collected from 16 plants. The thin gray lines in this trace are showing us how spur length on the y-axis is changing with day of flower collection for each of those 16 plants. Each line is a different plant. And the dark green line is the overall average trend for the change in spur length averaged across the 16 plants. As expected, spur length is decreasing from first to last flowers. And in fact, in the very last flowers to produce, they had hardly any spur at all, right? These values are pretty close to 0. Are spurs just changing with overall flower size in general? Is this just like a lockstep shrinking of flower size, or is there something special happening with the spurs in particular? To answer that question, we compared the change in spur length to some of the other traits that Rasheen measured. I'm just going to show you one of them here. Here on the y-axis, we're now looking at the ratio of spur length relative to the flower height, excluding the spur. And what you see is that for the first 12 or so days of flowering, spurs were shrinking in lockstep with the rest of the flower, right? There was no change in that ratio for the first few days of flowering. Even though spurs were decreasing, the rest of the flower was also decreasing at the same rate. But after about the 12th day, we start to see a more rapid decrease in spur size than we did in the rest of the flower. So that's suggesting that spurs are in fact under some independent control, and they're exhibiting a different pattern within plant variation than the rest of the flower, which is certainly interesting to us. We wanted to know whether there was any biological significance to that variation. And of course, because we're talking about nectar spurs on flowers here, the obvious thing to ask is whether it's affecting the way that pollinators are interacting with the flowers. So Rasheen's work also included some evaluation of pollinator visitation to these plants and how it changed over the season. The first thing we had to do was figure out who pollinates these plants at all, because there's no report of it in the Western scientific literature. And at least at this one population, over 40 hours of observation to Halenia deflexa patches, the most abundant pollinator we saw was Bombus species. So we saw 114 visits to Halenia plants, and 86% of those were by Bombus. So yeah, we figured out something about who pollinates the flowers, which was a good first step. We next wanted to know if the rate of visitation changed over the course of the season. I'll show you the data on our next slide. So here on the y-axis is the probability of a Bombus visiting a patch of Halenia during our 30-minute observation period. Each point here is a patch. It's a 1 if at least one Bombus came to the patch during that observation, and it's a 0 if we didn't see Bombus visiting the patch during that 30 minutes. The x-axis is the day that we were at the population observing pollinators for that observation. And what I'm sure you see is that the probability of a Bombus visiting a patch declined drastically mid-season. So after the August 6th visit, we no longer observed any Bombus visiting Halenia deflexa at all, despite the fact that there was still plenty of Bombus in the population. This change in visitation probability coincided with the timing that the plants were starting to show those flowers with almost no spur at all, right? So there's this intriguing correlation going on. We're seeing this change in flower presentation that's driven by within plant variation in spur size, and it's coinciding in time with when Bombus stopped visiting the plants. But I stress that at this point, it's just a correlation. We don't know that there's any causal effect going on there, and that's something I would like to eventually tackle. Does it matter if Bombus stop visiting Halenia deflexa? Well, it might if it affects the probability that a flower is going to have to engage in self-fertilization versus outcross pollen receipt. And so that same summer, another honor student of mine, Sarah McConnell, did some work to assess the mating system of Halenia deflexa and to try to understand whether the probability of self-pollination changes over the course of the season. The first thing Sarah had to do was to figure out if Halenia deflexa is self-compatible at all. And through some hand pollination experiments where she applied either self-pollen or outcross pollen, we determined that yes, this plant is highly self-compatible. It can absolutely produce fruit when it's fertilized with its own pollen. So that was check 1. Some ongoing work in my lab is suggesting, though, that the quality of seed produced is lower after a self-pollination than after an outcross pollination, but that work is in progress. We next wanted to know if the probability of a flower being self-pollinated changed over time when spur length was changing and pollinator visitation was changing. To do this, Sarah repeatedly manipulated pairs of flowers on Halenia plants using embroidery floss to keep track of which flower had which manipulation. On some flowers, she removed the anthers so that they could not self-fertilize. On other flowers, she poked forceps around in them, but didn't actually remove the anthers, so self-fertilize was still an option. That was called the handle treatment. And when we look at whether or not a flower matured into a fruit, depending on whether it was handled but not emasculated, or if it was emasculated, if there was an increased reliance on self-pollination as the season progressed, I would expect the distance between these two lines to increase over time, but it did not. So somehow, despite the fact that pollinators weren't visiting towards the end of the season, at least not during our observation periods, the emasculated flowers still managed to set fruit. I'm not really sure what was going on here. One possibility is that there was some night pollination that we simply weren't observing because this site is so hard to access at the nighttime. But I do have some ideas for experiments to try and get a grip on evening pollination. Another possibility is that maybe there's apomixis going on, but once again, that requires some further testing. All right, so there's a number of directions to go with this work. One, I want to test the causality of pollinator response. I'm running out of time, so I'm not going to go into depth on how. Two, I'd like to test the idea that if selection acts to increase the variation in nectar spur length or decrease the variation in nectar spur length within plants, can a plant population actually respond to that selection? And the way I plan to do that is through artificial selection on the coefficient of variation in spur length. And finally, if that selection is successful, I'd like to bring those selection lines back to the field to see how having increased or decreased variation in spur length affects fitness in a field setting. Is within plant variation a target of selection? I'm seeing some possible responses of pollinators to within plant variation in the field, but there's work to do there to show a causality of effect still. And second, I've got some ongoing tests to directly test whether selection is acting on within plant variation and whether plants can respond to that selection. All right, in my last few minutes, I'm going to tell you about an idea I'm just starting to dip my toe into. And this is the idea that maybe the variation in organ level traits expressed within a plant provide a blueprint for diversification of those traits when we look across a lineage. There's a pretty widespread and well-founded idea in evolutionary biology that adaptive evolution will tend to follow the path of least resistance. So if a trait needs to go from point A to point B on the adaptive landscape, it's going to follow the path of least resistance, which may not be the most direct path. One of the ways this pattern manifests in animals is shown in the figure here with really small axis labels, but that's okay. This is some work on side-blotched lizards, but it's repeated in other taxa as well, showing that if you take the entire clade of side-blotched lizards and measure several populations of species within those clade and plot the variability in a trait within species, within populations on the x-axis, so how variable is a given trait within populations, and then ask how much does that trait diverge across species within the same clade. So here we've got each point representing a trait. The X value is how variable is that trait within populations on average across a clade. And then the Y-axis is how much does it vary across species within the clade. There tends to be a positive correlation. So the traits that show the most variation within populations are the traits that are most involved in differentiation among species within a clade. That works in animals, but in plants, as we've already discussed, most of the variation in organ level traits is falling within the individual. And so I'm starting to think about whether the traits that are most variable within individuals are also the traits that show the most divergence among species. In Halenia, right, these are preserved flowers from machines work. We see these differences in spur length within individuals. Those differences in spur length occur either through changes in cell proliferation during flower development or cell elongation or some combination of those things. And so I'm also wondering, when spur length evolves across species, is it those same changes to cell proliferation or cell elongation that's responsible for the divergence among species? Because in genus Halenia, I'm showing you just 6 species here, there's a lot of variation in spur length and in spur curvature. And so I think this is a good system for trying to think about which traits are most likely to evolve during speciation, and whether within plant variation gives us some sense as to how that diversification among species occurs. So in other words, within plants of Halenia deflexa, do differences in spur length arise mostly from changes in cell size, cell shape, cell number, right? What's the floral developmental basis of the changes in spur length? And is that same basis the one that's manipulated during spur length evolution among species? I'm just starting to work on these ideas. I've got an honor student right now, Zach Brown, who's been imaging some of the flowers that Roisin collected to measure cell size and cell shape within the nectar spur, and then to count the number of cells within a nectar spur, right? So that will address that first part of the question. I don't have data for you yet, but that gives us a first sense of how mechanistically are we achieving changes in spur length within individual plants. And from there, I have a few ideas of what I'd like to do next. One, we could again use artificial selection, but this time try to select on that hard to select on trait, the mean spur size. And if we get response, I would predict that, the selection line that generates a smaller spur size is going to have similar developmental changes to the flower production to produce a small size as the changes that you see from flower to flower within a plant, right? So are those pathways that manipulate flower size within a plant the same ones that are employed to create an evolutionary change across generations? I'd also like to compare spur structure among those different species of Halenia to again ask, are these cellular changes that give rise to variation from flower to flower within a plant the same ones that are exploited to create diversification across the lineage? I don't have answers to this question yet, but I hope you agree it's kind of an interesting way to start thinking about within plant variation. Last slide, I promise. I know I'm kind of at the end of my talk here. I've been talking to you today about the potential for ecological and evolutionary significance of within plant variation. And I recognize that I don't have strong answers on any of the ideas I've put forth today. My work on the agent of selection ended up not providing evidence for my hypothesis, at least in the system that I was working on. And the work on the target of selection is kind of just in the first steps of getting going. Blueprint for diversification is even earlier still. But I hope that I've at least convinced you that it's worthwhile to think about the potential for ecological and evolutionary significance of within plant variation. If most variation in organ level traits is occurring within individuals, by at least considering its potential importance, we can hopefully gain a better understanding of how plants work, and it might help guide us to focus our attention on the traits that matter most for plant adaptation. Maybe within plant variation is actually a part of how a plant succeeds. With that, I've got a number of acknowledgements here, a number of honor students and technicians at Mount Allison University, people who helped me out at the University of Toronto, field sites for my PhD work and my current work at Mount Allison, and funding sources. Thank you all so much for your time, and I'd be happy to take any questions.